In this video, we will talk about the motivation and some additional background for our paper, Dilated Convolutional Neural Networks for Sequential Manifold-valued Data in Neuroimaging. Let us consider the motivation first. There are a number of reasons why sequential models for manifold-valued data are needed. Euclidean space is linear and simple. But manifold has nonlinear structure but is locally linear. The example shows a manifold where the samples are symmetric positive definite matrices. As we know, the distances between such matrices needs to take into account the geometry of the manifold, and Euclidean distance calculations are suboptimal. Creating a vector from a SPD matrix will not satisfy the manifold constraint. For sequential model, RNN is the default choice. But dilated CNN is making progress on speed and performance. Assume that we want to construct a sequential model for manifold-valued data shown here. One questions is which sequential model should we choose? In this case, we leverage recent development of dilated CNN. Normally, we will use an RNN model as our sequential model of choice. However, RNN has limitation in terms of running time as well as a phenomena known as gradient explosion during the training process. Here, we use a dilated CNN to avoid those disadvantages. Let us see what dilated CNN does. Imagine we are given x0 to x8 measurement in one sequential sample. At the d equals 1 level, we perform a standard convolution and derive the input to the hidden layer. But at d equals 2, we use a dilated factor to expand the field of view. And we repeat this process incrementally until we get to the output node in the network. Similar to the regular convolution, weights are learned by presenting the model many samples and back-propagating. While the technical details are described in the paper, let us look at the application motivating this work. In brain imaging applications, we often encounter sequential data, which may correspond to manifold-valued data, where standard sequential models may be suboptimal. In diffusion imaging, we acquire diffusion measurements from individual spatial locations inside the brain. A procedure called tractography can be used to estimate the structural connectivity between distinct regions shown here. The diffusion measurement at a voxel is related to the structural strength of pathways passing through that voxel. Our goal is to analyze these structural pathways in the brain across clinically disparate groups. We will do this by analyzing individual fiber bundles and asking if they are different across groups. Going from one region to another along a fiber bundle, we encounter a set of diffusion tensors which are 3 by 3 matrices that are also SPD. This is why we model each fiber bundle with a sequential model where the sequence corresponds to going from one region to the other. Our goal here is to analyze data acquired as part of an Alzheimer's disease study, which is a progressive neurodegenerative disease and is the most common form of dementia. It is believed that in AD, due to neurodegeneration, the fiber tracts in brain degenerate, leading to loss in structural connectivity. Biomarkers are related to onset of Alzheimer's disease. For example, amyloid pathology is measured via PIB imaging. And separately on the genetic side, the APOE4 gene is a risk factor for Alzheimer's disease. So we perform an analysis based on these two grouping variables. Here is an example of diffusion-weighted MR image and results from a tractography procedure showing structural connectivity between brain regions. For math part, we will discuss the operators on manifold. We used weighted Frechet mean to do convolution on manifold-valued field. The top is the one-dimension convolution. It can be viewed as a weighted sum. And the bottom is one-dimension weighted mean. The weighted mean has only scalar times difference from the convolution. So in dilated convolution, essentially we can cover the entire sequence with few depth. And the difference between dilated convolution and causal convolution is exponentially increasing as the number of layer increases. And we used an efficient recursive algorithm with theoretical guarantees to compute weighted Frechet mean. This algorithm works as a recursion where at each step it updates the weighted Frechet mean with the previous weighted Frechet mean and the new data sample. The recursion starts with the initial WFM as x0. The recursion update uses analytic expression of the shortest geodesic. We can define residual block by doing weighted Frechet mean of the output y and the input x with a learnable w. Notice that, with W being the two extremes 0 or 1, the output is Y or X, and with a fractional W, it will output a combination of X and Y. Thus, this block has flexibility to use or ignore the residual block. The last part is the statistical background. We have done permutation testing on the parameter space instead of the sample space. For permutation testing, we have two groups from biomarkers or APOE gene. We can calculate the distance between two groups in parameter space. Then we shuffled the samples and gave them the fake group. Similarly, we calculate the distance in parameter space. We repeat these operators to obtain the null distribution of the distance. If the original distance is large enough, we can conclude that it cannot happen by chance. We have performed group analysis using different biomarkers. We can achieve higher classification accuracy on the UCF11 data over the SOTA methods. In brain imaging applications, we have detected several fibers which can capture the group difference with statistical significance. Our proposed method is faster than the SOTA methods for both group analysis and the video classification experiments. Thank you for watching this video.